Well, thank you for that introduction, and thank you for inviting me here. I wasn't sure what to expect, I have to admit, when I came, and I usually go to these meetings that are sort of these broad evolution meeting or integrative and comparative biology meetings. So this felt very specific. But I think we've all sort of learned over the last few days just how variable everybody's research is and our research interests. And I've really appreciated and I've been excited by learning how other people are looking at traits both ecologically and evolutionarily. And my work definitely fits within this sort of broad idea of trait-based approaches to ocean life. But as Ken said in the introduction, my perspective is a little bit different. I'm actually interested in the traits themselves and how they evolve. And specifically myself and my lab, we're macroevolutionary biologists. So we're particularly interested in how interspecific trait variation evolves over time and what drives that. And specifically, we're really interested in morphology and morphologies that are relevant to ecology. So the traits that mediate how the fish or any organism interacts with its environment and with other conspecifics or more distantly related individuals. So today I want to tell you about a large project that has been sort of ongoing in my lab with some collaborators of mine on teleos fish body shape evolution. So teleos fishes, there's around 28,000 to 30,000 species. And so that accounts for maybe 50% of the entire vertebrate diversity on Earth. So they're sort of an important part of vertebrate diversity. And they have these really inspiring, to me, diversity of body sizes and shapes, all the way through, from these sort of very elongate forms through to these very wide-bodied forms, very deep-bodied. And then, we have the weirdos like the seahorse, which, quite frankly, I still struggle to believe is really a fish. And so we have this really sort of wonderful amount of diversity. But how do we go about trying to understand what has driven this diversity over time? And we know as sort of evolutionary biologists that sort of any body shape that enhances fitness, so, either increases survival or increases reproductive output, can be acted upon by natural selection. And that means that sort of any of these performances can be really important, things like locomotor performance, mate attraction, prey capture performance. And the precise sort of performance and thus body shape that is going to enhance fitness is going to be highly dependent on a huge amount of sort of interacting factors about the fish's biology and its environment as well. So for example, locomotor performance might enhance fitness by allowing you to better catch your prey or escape predators. But the precise locomotor performance that's going to enhance fitness is going to depend on where you're living. If you're in an open habitat, then maybe being able to swim very fast in a straight line is good. Whereas if you're in a very complex habitat, there's a lot of obstacles, so maneuverability. And so different body shapes are going to enhance fitness depending on this sort of wide variety of different factors. And so what we're really interested in doing is trying to understand how these different things ultimately influence the diversity of body shapes we see today. And to do this, we need three things. Obviously, first of all, we need traits. That's why we're all here. And for me, I collect a lot of the morphological data, but I then also need other kinds of trait data as well. We need ecological and environmental data to be able to sort of look at the drivers. And with fishes, we are really lucky to have FishBase that has a huge amount of ecological data that is a community effort. So that's really important. And then, of course, we have all these wonderful sort of big environmental data sets as well. So we're sort of integrating multiple trait databases to be able to do this work. And also here we have genetic data. So we need a phylogeny. That's the second thing that we need. So we need genetic data to be able to build our phylogenies. these phylogenies are really important. So when they're time calibrated, our branch lengths are in millions of years. And this allows us to know the timing of divergence of these species and the relationships between the species. So we have the tree, as I'm showing here, and we have traits. And we've got both sort of categorical traits, as I'm showing with the color, and then we can also have continuous traits, as I'm showing sort of with the size and the shape here. So how do we bring these two things together? We need a model of evolution. So we have a model of evolution that shows how the traits evolve through time, and that's along those branches of the phylogeny. And we have different models for different types of trait. We use continuous time Markov models for categorical traits. And for our continuous traits, and that's what our morphology is all continuous. In general, we're using these generalized Ornstein-Uhlenbeck models. These are an adaptive model. So we have the primary adaptive optimum given by theta, and the pull towards that peak is driven by alpha. And then we have sort of a variance parameter here, which is sigma. And so we have these three traits, these three parameters that we can estimate. And if alpha is going to be 0, then we lose the adaptive part and it just becomes a random walk or Brownian motion model. So this is how we're sort of modeling trait evolution along the phylogeny. So that's sort of the general approach that we take to doing this. And I now want to sort of get into specifics about this project. So I first want to talk today about how we went about quantifying shape diversity and what we found. Then want to do a little bit, step back a little bit and talk about how we collected this data using undergraduate researchers and how we brought them into helping us collect our data and then also being part of our scientific discovery process. And then I want to illustrate what we've been able to use this trait database for and give you a couple of examples of projects that we have worked on. So we want to be able to quantify shape diversity. I can show you pictures and we know it's diverse, but how do we actually go about putting some numbers on that? So we went to the museum, we pulled fish out of jars and measured them. And we wanted to measure a lot, so we couldn't take really detailed measurements. So we came up with this set of traits that sort of capture the overall shape of the fish on sort of lengths, depths, and widths of the fish. And these are all sort of very standard measures in ichthyology. And we know that they are related to the way the fish swims, the way it eats, where it is in the habitat, and that sort of thing. So they're sort of functionally relevant. The other thing that we also have here, which I'm not showing on the picture, is we had the first and the longest spine in each fin. These are really important sort of anti-predator traits that we're interested in as well. So we took these measurements. We also have detailed lateral photographs, so we can actually collect more detailed data on this sort of image here, but we don't have any width information with those. So we've collected these traits. We spent our time at the Smithsonian Museum. We spent 7 months there over 3 years, and we had a team of 5 to 6 people, including our undergrad researchers. We camped out in the corridor, in the collections, freezing, smelling of fishy ethanol. You know, it was a great experience for all. Look, everybody is happy, so it's not too much an issue. Although after a while, I spent 2 months there and I did go a little bit crazy. But I think this was all sort of worth it because in the end, we were able to measure over 16,000 specimens. And this encompassed around 6,000 species. So we have a little bit of intra-specific variation here, but not really enough to look at it on its own. It's more there to just feel we have a good estimate of the species average. And we have a very broad taxonomic sample. We've got around 90% of extant families and 96% of living orders. So this is around a fifth of the entire diversity. So not as big as some. I thought yesterday when I was blown away by the 1 billion images, I was very proud of this data set till I saw that. But we have to open all the jars ourselves, so I suppose maybe. But so we have now this huge amount of data. And the first thing, we're particularly interested in shape, but we know we've had a lot of conversations about size, and size is really important too. And particularly if we're interested in shape, this is our smallest fish in our data set, and this is one of the largest. How do we compare shape when they vary so much in size? We really need to take into account size. And we've been talking a lot about how size is this sort of fundamental trait. And I think that's interesting because one of my questions is actually, what is size? Like, how do we measure size? And that might seem silly, because we know that this is smaller and this is bigger. But in traditional sort of ichthyology, we use a length. So either standard length, which is sort of tip of the snout to just by the before the caudal fin, or total length of the fish. And, you know, here I've got standard length plotted against all our other body traits. And you can see, and this is a line from a phylogenetic regression, and this is close to isometry. This is close to a 1 line. But you can see that while there is a very strong relationship, there's also a lot of variance. And you'll notice, particularly sort of in this area here, we've got a lot of variability. And that's these elongated forms of fish. They're just so very long that they're so different from everything else. And that sort of brings up this important point. You know, when we use just this sort of standard idea of size, we've got the same length here, but I think we'd all agree that these are probably very different sizes of fish. And so what do we do about that? The obvious thing, if we had freshly caught specimens, would just be to use body mass, right? That would be a simple way around. But these are collections fishes, and they're sort of degraded. They've been used for other studies. A lot of them didn't have anything inside their body cavity. Some of them had metal tags. And so sort of weight, we didn't feel was sort of very good. So we also considered that potentially using the geometric mean of our 3 sort of major dimensions. So standard length, maximum body depth, and maximum body width, as that would sort of encompass those 3 dimensions of size rather than just length. So there are multiple ways that we can measure size. And then I want to be able to control for size so I can just focus on shape. And there are multiple ways of doing that as well. And it all depends on whether you want to just control for isometry. So that's when a larger individual is just the smaller individual scaled up. So sort of a perfect representation, but just bigger. Or whether you also want to include allometry, which is where the dimensions are predictable from size, but they're not identical. And so if you only want to remove isometry, you can use log shape ratio. So you divide your trait value by size and take the log of it. Or if you want to remove both of these effects of size, you can take the residuals from a phylogenetic regression of your size versus the trait. And so this will remove both of these things. And so as I'm going to illustrate now, the different methods of estimating size and of controlling for size can actually have a strong impact on what we identify as the primary axes of variation. So here's our trait space using phylogenetic residuals and standard length. And this is our first principal components axis that I'm illustrating here. So this describes an axis from these very deep, wide-bodied fishes, so like these sea devils and frogfishes and things, all the way through to these extremely elongated forms. And you can see it's explaining around 45% of the variation. What we have on our secondary axis then contrasts mouth size to caudal peduncle. So that's right by the tail fin here. And so we have these very small mouths, very wide caudal peduncles up here, and then these very big mouths and tapering caudal peduncles down here. And you can see here from the sort of 3D density plot that nearly every fish is very tight within this area of region of morphospace. And the variation on PC2 is only within the elongated forms. So everything is very sort of tight up here. This changes somewhat when we use our geometric mean and our log shape ratios. So now our PC axis explains around half the amount of variation. And instead of being sort of the elongation axis, it's actually a width axis. So we've got these very narrow, laterally compressed bodies like these flatfishes here. And then we've got these very wide dorsoventrally flattened bodies over here. And then our secondary axis then is more sort of elongation, contrasting length and depth. So we've got these very deep-bodied forms and these very elongate forms up here. And this PC2 explains around 20% of the variation. So really what has happened here is this has sort of decomposed that first primary axis of the previous one where we looked at standard length and using the phylogenetic regression. We've sort of split that primary axis into two this way. And then we've split out width and then length and depth up here. And you can see we've got a much broader peak, so that there's sort of a much broader area in which fishes are occupying this trait space. So I think this is really sort of interesting because it sort of illustrates the choices we make about how we measure size, how we correct for size. That can really impact sort of downstream analyses. So our major axes of evolutionary variation that we identify really depends on which method we use. We use the phylogenetic residuals and standard length, elongation, and a contrast in the expansion of the head and the caudal region is our sort of primary two axes. Whereas if we're doing our log shape ratios and the geometric mean, we've got width and elongation as our primary axes of variation. So I think that's just sort of an interesting illustration of having to think about our decisions when we're doing our analyses. And so now I just want to go on and sort of do a little bit of an aside really about sort of integrating our undergraduate researchers into this project. So we knew we had an awful lot of data to collect. We didn't have enough people to be able to do that. And so we really sort of thought that undergraduate researchers were the way to go. And I think this is true for quite a lot of studies that have a large amount of phenotypic traits. If we want to add in another omics word, we could call it phenomics, measurement and analysis of large sets of traits. And usually collecting the phenome data is often a rate limiting step unless you can automate it. And we saw some beautiful examples of that automation yesterday, which are just amazing to me. But we couldn't have that automation, so we went for undergraduate researchers. But we also had a lot of these undergraduate researchers. So that was really sort of too many to sort of mentor individually on projects. So we built this course-based undergraduate research experience where we had a cohort of students sort of all working together. And so we had these sort of two aims for this project. The first is practical, right? To train our students to collect the data and identify suitable candidates for the paid summer intern researchship at the museum. And I just highlighted it's paid. I think it's really important to pay our students during the summer because it's a really important sort of equity issue. So we had this practical aspect. We wanted to have good data collectors, but we also wanted an educational component too. And so we really wanted the students to be able to participate in scientific discovery and experience the entire process of science. And we had a massive team for this project. All of those here in this sort of bluey color, these are all our undergraduate researchers. This is my co-PI, some grad students and postdocs, and some grad students and lab techs here. So we have a large number of people. And these come from like 3 different cohorts of undergraduates. And we had these sort of 4 sections that we went through with our students. And we told them overall their goal was to develop and test a publishable scientific hypothesis. So there was certain pressure on them to really make sure that the ideas themselves we could foresee being a publication. Obviously, there's a lot of vagaries that go on with that, but we were sort of holding them to that standard. And we'd split it up into these sort of 4 different sections. First, we did the phenotyping training. Then there was actually a break here. This was the summer, and then we took the students to the museum. And then they did these 3 other sections of hypothesis development, training and analysis, and then finally interpreting and presenting the results. And this happened over 18 months. So often these cures will just be sort of a normal lab for a semester. We did this, so we only met for maybe an hour and a half each week. that meant everybody could do this sort of on top of their normal work. And it sort of increased our diversity of students in terms of those that could actually afford the time to be able to do this. And like I said, we had these 4 sections, and we were really focusing on these 3 after the phenotyping training, because we think that these are things that often our undergraduates don't get to experience in traditional sort of educational settings and lectures and labs. But they are really critical for sort of modern scientific discovery. So focusing on hypothesis development, learning how to analyze data, and then present their results. And so I just sort of want to walk you through. And the reason I did this is that I used to just very briefly mention this. And then I'd have so many people come up to me afterwards asking about the undergraduate research that I've started including this into my talk. So hypothesis development. We first, of course, had to make sure that they had the skills that would enable them to come up with some good scientific hypotheses. So we sort of made sure they were comfortable reading papers. And because we're doing macroevolution, that sort of scale of thinking is often quite hard for students. So we wanted to get them comfortable sort of reading microevolutionary studies and being able to sort of scale up the expectations there. So we gave them experience with that. And also how to find useful papers. Once we felt comfortable with that, we sent them off into the literature, finding papers that were relevant to how body shape evolves in fishes, and they would sort of submit their hypotheses. And one thing that we found was really important was this anonymous list. They would send us their hypotheses. We would then sort of curate them, put them in order. And this was really important for 2 reasons. Obviously, first one is they don't know whose idea it was. So they're not critiquing a person, they're just critiquing an idea. And the second was that we found that we as mentors could add ideas without biasing the class. Because as soon as you suggest something, they're like, oh, well, the professor suggested that, therefore that must be better. So we could add things that they'd sort of missed, and they wouldn't be biased by that. We could just go through and critique all of them. And so that turned out to be really important. So once they had all these sort of hypotheses, we'd go through, critique, identify which ones were possible, which ones we had data for, how long each would take. So we'd sort of narrowed it down and picked a particular project for all of them to work on. We then sort of trained them to use R. And so I don't know if you've had this experience, but for me, when you're trying to teach biologists to do any form of programming, they sort of freak out and just don't want to think about that. And they're like, I'm useless with computers. I can't tell you how many times I've heard that. So one thing we found helped was to use the real data, not necessarily all of it, but they could actually be starting to make discoveries along the way. So that way they really were understanding the connection of why they were learning to do this, because they could actually start making discoveries while they were also learning. So really emphasized to them why this was important. The other thing that we found was a lot of people, they think they're useless with computers. So the first time they make a mistake with their code, they're like, that just proves it. Whereas I don't know if any of you have tried writing code up in like this on the board and trying to do it. You always make mistakes. And that was really helpful because they could see we were making mistakes and that was just a normal part of coding. And so that was very helpful. And they could help us debug and point out that we'd missed a bracket or whatever. And we also focused on visualizations. It would be lovely to go really in depth, but we just didn't have time to give them a full statistics class. So visualizations are really good because they can really start identifying patterns. But we did show them how to do these sort of complex phylogenetic comparative methods as well. It's just that our emphasis was on visualization. Computer just had to think about transitioning for a minute. So then we did so once they learned how to do the analyses, we then gave them the whole data set that they could run their analyses on and start sort of interpreting the results. And the one thing that we really did was encourage them to present at local conferences that would actually allow them to invite their friends and family, because they'd been working on this for 1.5 years now, and their housemates were wondering what they were doing, looking at sort of dead fish the whole time. And it really allowed them to sort of show people what they've been working on. And we allowed the students to choose whether they wanted to do a poster or a talk, and they could work in small groups if they wanted to, or on their own, so whatever they felt most comfortable with. And the feedback that we got from the students, this really was the highlight of it, that they really sort of enjoyed being able to share this with their friends and family. And for us, it was really great because this is where we could really see the students taking ownership of the project, and they could really see what they'd achieved in the 18 months. And we've had so far up to 16 talks and 5 posters, both at sort of local conferences and more recently at the International Conference for Integrative and Comparative Biology. And that's because of the pandemic. Now we're recording talks. We could actually have multiple undergraduates give the same talk, which you couldn't have at a conference. So I suppose that's our way of sort of making lemonade from lemons of the pandemic. And the hope was that all of this allowed them to really fully experience the scientific process in all its sort of messy gloriousness, right? That they realizing it's not this simple linear thing. You find something out, that doesn't make sense. You have to go and figure out what data you need to collect to understand it. And so really getting a good feel for really what science is. So we did this to enable us to be able to collect our data, really. We wanted to give our students more than just the experience of collecting the data, but really being part of the science as well. And so now I want to move on to give some examples of the science that we've been doing with this data set. So I'll just remind you, we have all of these different particular factors that can influence body shape. And initially, we've sort of pulled out individual ones to look at them sort of one-on-one. And our predictions of how shape may influence fitness and how it may evolve come from sort of 2 major sources. And here at my title, Macroevolutionary Signatures of Microevolutionary Processes. That's really what we're looking for, right? We're trying to see if we can see patterns within the diversity of body shape traits that make sense based on what we know of microevolutionary processes. And so we sort of have 2 ways of making predictions. The first is from these biomechanical models that link shape to performance. So these are based on sort of principles of physics of how shapes may optimize certain locomotor or feeding performances. And then a lot of them come from sort of these intra-specific experiments or observational studies of how shape changes relative to various differences in habitat or environment. So looking at temperature or dissolved oxygen or presence of predators, etc. So I just want to illustrate both of these 2 things with one looking at a project on habitat complexity, and then the other looking at maybe the coevolution of anti-predator defenses and body shape. So I'm going to start off talking about habitat complexity. This actually was a project from our CURE. So these are the undergraduate students that worked on this. And I think they really liked these biomechanical models because it provided these very sort of strong physics-based predictions. And the one they chose to work on is a really sort of well-established trade-off of swimming in fishes. So the common expectation within the fish literature is a trade-off between steady swimming, so that's sort of fast straight line swimming, and unsteady swimming, which is changes in direction and velocity. And if you're good at steady swimming, you can't be good at unsteady swimming because the shapes that allow you to do that are very different. So for the steady swimming, we have these very fusiform, so highly efficient swimming shapes that are very streamlined. Whereas for unsteady swimming, so being highly maneuverable, you get these deep bodies and these deep caudal peduncle regions, which maximize thrust and maneuverability. And so when unsteady swimming performance maximizes fitness, we're expecting the evolution of these sort of deep body shapes. And when steady swimming performance maximizes fitness, we expect the evolution of these sort of fusiform and highly efficient shapes. And so one of the expectations here is this is linked to the habitats in which they live in. If you're in a complex habitat, we expect that to select for unsteady swimming, and open habitats to select for steady swimming. So the first issue that the students had to sort of deal with is, well, how do we go about measuring differences between habitat? And, I'm sure everybody here is aware there are these sort of very continuous measures. We can look at sort of rugosity, but, we're trying to do this for a large number of fish species and sort of get an average for that species. So that sort of data is not available for everything. So we needed sort of a more simple measure. And we decided that we would start off really simple and sort of hopefully in a way that nobody could argue with us that reef habitats are much more complex than any other marine habitat. So if we just simply divide our fishes into those that live on reefs and those that live in non-reef habitats, people should feel fairly comfortable that this is a difference in complexity between these two habitats. So we focused just on marine species. So our data set is now around 3,500. And so we wanted to see whether fishes living in reef habitats had these body shapes that were suited for unsteady swimming. And similarly here, much more sort of efficient fusiform shapes in the non-reef habitats. So we were able to get that information for all of our marine species. And you can see here that there's been multiple invasions of reef habitats across the Teleos phylogeny. So that's here in blue. So we've got sort of multiple invasions. And this is important because this provides our degree of freedom to look for an association between shape and the habitat. So if there was only one of these, this would be problematic because you have a single degree of freedom. That's just a single coin toss. So we need multiple of these. And I'm just showing here one possible history. This is actually a Bayesian method. So we integrate over uncertainty and we use multiple different maps. So we're looking here at fineness ratio. This is used in aircraft and other things to describe how sort of streamlined the shape is. And it's just calculated by the standard length here divided by the square root of the maximum body depth by maximum fish width. So this shows how streamlined they are. So here we've got sort of the optimum streamlining of sort of a tuna. Anything above that is sort of much more elongated form like this pipefish. And anything smaller than this, anything lower than this is the sort of more deep-bodied fishes. And so you can see there is, on average, a difference that these reef fishes are less streamlined, just as we predicted. And if we separate out and just look at body depth and the caudal peduncle region, we see exactly the same thing. So these reef fishes have deeper bodies and deeper caudal peduncles. And you might have also noticed here these sort of very low R-squared values. So that obviously means there are many other factors influencing shape, but we know at the scale across all the Teleos fishes that habitat complexity is not the only thing influencing that. We know it's a complex interaction. And so it's not surprising that it's low. And in fact, for me, I was actually surprised that we could see a signature of this using these sort of very rough distinctions between reef and non-reef living. But just to sort of summarize, what we were able to show is that these biomechanical models that we know sort of work on the basis of physics actually do predict some of the patterns that we see across all of Teleos. Fishes that are living in open habitats have these streamlined bodies, whereas those in the complex habitats have these deeper bodies. And, this is very simplified, like I said, and there are many other things influencing shape, but I think this still is a nice illustration of how we can see these signatures of microevolutionary processes at a macroevolutionary scale. And so now I want to go on and provide another example of the kind of work that we've been doing, and this is looking at anti-predator defenses. And I'm afraid here, this is not just marine. This is going to include both freshwater and marine fishes, so please don't hold that against me. So the first thing to know is that the majority of predators that feed on fishes are gate limited. So that means they have to be able to get the fish whole into their mouth to be able to feed on it. And in general, predators are pretty good at choosing which fishes are going to fit into their mouth. And that's because often it can be quite costly to make that mistake. So we've got here a picture of a pike that was found dead with this other massive fish lodged in its mouth. So in general, predators are pretty good at choosing fishes based on whether they will fit into their mouth or not. And intraspecific experiments and between population comparisons have consistently found two responses when fishes are exposed to predators. The first one is that fish always seem to increase their relative body depth in response to predation cues. And these are just some of the fishes that this has been found to be true in. And these are found across the phylogeny. They're not all closely related. The expectation here is if you're increasing your body depth, you're exceeding the gape of the predator. So the predator has to have a larger gape to be able to eat you. As well as we mentioned in the coral reef example, these deeper shapes also sort of help with acceleration, turning rate, and maximum speed achieved while maneuvering. So this potentially has sort of two benefits to escape predators. The other one that we see frequently is an increase in armor plus the number and length of the fin spines in response to predation cues. And so we can see here, a lot of fishes will have these pretty gnarly spines in their dorsal fin, and their anal fin, and sometimes even their pelvic or their pectoral fins. And so what's going on here is you're increasing the handling time. So imagine trying to eat a very spiky burger. It's going to take you a lot longer. And that's the same for any fish eating another fish that has spines. And that your burger doesn't have an inclination to run away, but the fish does. And so it gives it a higher chance of escape. Plus, the predator also is risking injury when they are attacking a fish with these spines. And the one study that has looked at these two things in conjunction shows that they do actually work together. So they don't just increase body depth or increase their spines, but just at least within this pumpkin seed sunfish, when they're exposed to predation cues, they increase their body depth and they increase their dorsal fin spine length, but not their body length. So they're not just increasing size overall of everything. It really is the depth and the fin spine length that are increasing. And I think this makes sense because there's synergy between these two things, right? Fin spines and relative body dimensions can work together. So this fish relative to this fish is going to fit into more predators' mouths. So there's a benefit here. But if you put a spine on, then it's an even bigger gape that is needed to eat you. And probably the energy investment in making the spine is not as much as making a deeper bodied fish. So we might expect synergy. And we can actually sort of start to test this hypothesis, as some fishes have spines just on their vertical axis, in their dorsal and their anal fins, and others have spines on their horizontal axis. So if there's synergy, what we expect to see is those with spines on their vertical axis and their dorsal and anal fin, they will get deeper bodies, while those with their spines in their pectoral fins, so on the horizontal, if they're working together, they should get wider bodies. So we can actually start to look at this, and we're using these generalized Ornstein-Uhlenbeck models. Here we're specifically interested in theta, that is the primary optimum. And so this is going to show how the presence or absence of spines influences both body depth or body width. And the influence of phylogeny and anything else that could be influencing depth and width are going to be captured by the alpha and the sigma parameter. So we use a model fitting approach. So we either fit a single theta across all of the fishes or allow it to vary based depending on the presence and absence of spines. And so by doing that, we can look to see whether body dimensions are dependent on the presence of fin spines. And then by looking at the absolute values of theta, we can look to see whether spiny lineages have deeper and wider bodies. So what we find, so this is looking at body width. So we've got the primary optimum on the x-axis of body width. And in the color orange, we've got this, they have no spines. And then in the blue, the spines are present. So here we've got fishes with pectoral fins. The optima for body width is much higher than it is for any fishes that do not have spines in their pectoral fins. Whereas if they've got spines on the vertical axis, so this is going against the body dimension we're looking at, which is body width, there's no difference in the primary optimum. It's highly overlapping. So this is exactly as we expect if there's synergy here. And we see the same thing when we look at the dorsal and anal fin spines and body depth. Those with spines on their vertical axis have deeper bodies than those that don't have spines on that axis, but those that have the pectoral fin spines, there's no influence on body depth. In fact, the best fitting model was just the universal theta model. So spines have no influence. The presence and absence of pectoral fin spines have no influence on body depth. So this sort of supports evidence of synergy between the fin spines and the relative body dimensions, suggesting that predation could be driving these sort of patterns that we see at these large scales. Obviously, we can't prove that, but it's very suggestive because we know that these things happen on a smaller microevolutionary scale, and we see these same patterns at the large scale too. But I've hidden something here a little bit, is that pectoral fin spines really have primarily just evolved once in catfishes. And so that means that we didn't have many catfishes in that original analysis. In fact, we had probably about 10. And so really, we need to better understand the relationship between sort of pectoral fin spines and body width. And we now have around 500 species of catfishes. Some of them have lost their spines, others have retained them. So we now have sort of multiple comparisons that we can look at again. And so in the past year, my undergrads got interested in this, mainly because they found out that catfishes have venom, and that's awesome. And that's another sort of anti-predator strategy. So they got sort of really interested in the coevolution of these different anti-predator strategies, body dimensions, spines, and then also venom, which is associated with the spines themselves. So these are the four undergrads. You can see this is the Zoom generation that we're living in right now. And they had these two predictions. The first, based on our previous work, was that these body dimensions will follow the fin spine orientation. But then they also were thinking about, because multiple lineages have lost both spines and venom, then the smaller species, they thought that the smaller species are less likely to have lost them because they're going to be more vulnerable to a lot more predators. Because they're smaller, they can fit in the gapes of many fishes. So they thought that smaller species are less likely to have lost their defenses, and they wanted to look at that. And so this is just an illustration. You can see that the presence and absence of venom varies across the phylogeny, as does the presence and absence of all spines. If you split this out to dorsal and pectoral, there's more variation, but I just lumped them in here to illustrate that. So within catfishes, we find exactly what we were seeing in our previous analysis, that there is a relationship between body depth and fin spine length, and this is controlling for phylogenetic relationships. So deeper catfishes have longer dorsal spines, and wider catfishes have longer pectoral spines. So that sort of supports what we were finding previously, but those species that were smaller had not retained their anti-predator defenses. And this was sort of surprising. We think, smaller fishes should be more vulnerable to predation. So what's going on here? Well, with catfishes, it turns out that these smaller catfishes, they probably evolved other strategies that enable them to avoid predation. So this is the sort of infamous kangaroo, where you hear those nightmarish stories of people swimming in the Amazon and then having to get the fish extracted from their urethra. That's not their natural habitat. It's actually the gills of other fishes. And so if you're in the gills of other fishes, you're probably not really exposed to predators individually. And so potentially there might be sort of a trade-off, at least in terms of sort of investment in anti-predator strategies going on here. But what we do have is a confirmation of the synergy between the spines and relative body dimensions. And so these trade-offs is something that my colleague Jen Hodge, she's now my postdoc, has looked at within butterfly fishes. It's sort of the investment and sort of trade-offs between predator avoidance and defensive traits within fishes. And she's working on butterfly fishes. So of course, the first thing that we did with this was to say, okay, let's look at this relationship between body depth and fin spines on the vertical axis. Because we've seen this in all these other groups. Actually, we don't see it in butterfly fishes. So we thought this was universal, but it's not. And so actually deeper bodied butterfly fishes don't have longer dorsal and anal fin spines. And we still need to sort of delve into why this is. But butterfly fishes are exceptionally deep bodied. They're in the 97th percentile of relative body depth of all fishes. And so maybe they're just so deep bodied at this point that they don't need to sort of keep increasing their depth through their spines and their body depth. But they do have this sort of interesting investment choice that these butterfly fishes that have weaker defenses, so we've got here the optima of the relative dorsal fin spine length and the optima of the anal spine. So these are sort of weaker defenses down here because they have smaller spines. So those with these sort of weaker spines have less exposed feeding strategies. So they're facultative grazers or obligate grazers. So they're living in the reef and around the corals. They're likely to be much more protected from predators than these benthic hunters that have these much more robust spines. These are out coursing, looking for food, and so much more exposed to predators. So from all of this, we can sort of summarize that we get synergy between fin spines and relative body dimensions, but not in butterfly fishes. And there are likely to be trade-offs between predator avoidance and defensive traits at this large scale. We've seen that in butterfly fishes and potentially in catfishes, but we need to drill down further. So I've given you a couple of examples of what we've looked at, and we've looked at a variety of different things. We've looked at how swimming mode influences it, how water depth influences shape, position in the water column influences shape. But these are all sort of looking at things individually. And we know, as I've illustrated here, that this is sort of a much more complex interactive network. And so what we're working on is sort of increasing the complexity of these models, being able to include all of these sort of multiple predictors. And the other thing that I'm really interested in doing and why it's been so great to be here and be inspired by you all is to sort of include the community context. We're ignoring that these traits are actually evolving within a community. It's not a vacuum. It's the traits that they can evolve are also going to depend on competition, what other traits and other species are already there, and also on predation. And so that's something that we're working on, is trying to sort of incorporate the community context into this in the future. So just remains for me to thank you all for coming here and for listening, and I'll take any questions.
Thank you, Samantha, for a really impressive breadth of insights from a beautiful data set.
Thank you.
It looks like Jessica. I have a couple questions, but we'll let Jessica. No, that's okay.
This is so fascinating. I actually have a bunch of questions, but I'll just restrict myself to one. I'm kind of curious if you have any data on differences in life history stages, the adult versus juvenile versus larval, because there can be, quite a few examples that, I even can think of very distinct morphological differences between different life stages, like mola or eels or.
Yeah, absolutely. So we made sure to have adult-sized specimens. So this is all within the sort of adult size. But yeah, it's going to be completely different for juvenile fishes, and that would be another way of trying to expand this, is to look at how that changes over ontogeny. It would be really interesting. There's one behind you. Okay.
Hi, yeah, thank you. That was really interesting and also beautiful, that talk. I'm not as familiar with fish studies, and so I was just wondering, in phytoplankton ecology, we think a lot about morphologies as well. And in setting up your coevolution of anti-predator defenses study, you were saying that the fish have evolved spines and body depth, I think that's the correct term, in response to predator cues. And I was just wondering how that information is known, whether there's actually like an evolution study on a fish which has such a long lifespan, or is it like a correlation? And just how is that information determined?
Yeah, so various studies have shown a plastic response, so an environmentally driven response sometimes to this, which over evolutionary time can sort of become canalized, at least theoretically. But other studies have shown between population differentiation that's heritable, where there's populations that have predators and those that don't have different traits, either spine length or body depth. So both those kinds of studies exist.
Hi, I had a question about if looking at fish and more in an individual fish versus a schooling type of fish, and if you saw any differences in defense traits along that axis.
Yeah, I mean, that is a great question. It's definitely, one of the theories would predict that schooling is different. It's actually pretty hard to get good data across all of our species on really which ones are schooling and which ones are not. I had a master's student that was actually interested in pattern evolution and so that again, we're trying to get the group size information to look at it as an anti-predator strategy of pattern plus these defenses, but we're not there yet. But yeah.
While I'm walking over, I'll ask my question about fish, which I saw a few times flash by some information about deep sea fish. I wondered how represented mesopelagic and bathypelagic forms were in your data. I was struck by like the example you showed early on of the relative rarity of big mouth, small tail forms, and I wondered if they're just missing from the collections.
So we do have, obviously we're limited by what was in the Smithsonian, but it's the largest fish collection in the world, unless Paris slightly is one of the two. And they did have a decent number of deep sea forms. So we were able to actually look at the evolution of deep sea forms with the amount of data that we had. And we do find that they're sort of evolving these pretty unusual forms that people have suggested previously, that they're large mouths and sort of tapering caudal peduncles. And so obviously there aren't that many deep sea fishes, and I think the proportions we have are fairly representative within the data set.
Yes, we often look for adaptive features, but did you have any ability to find traits that were neutral or maladaptive that, because of the genetic evolutionary process, are still within groups of these fish?
I don't know that we would really have the opportunity to look at maladaptive. I mean, we certainly, when we're doing analyses, we will certainly throw traits in that we're not expecting to be influenced by our factors. So when we looked at those reef fishes, we were expecting traits very strongly linked to locomotion, but not feeding. And so when we put our feeding traits in, they weren't influenced by whether they were reef or non-reef. So we sort of can validate that some traits that we are predicting to be affected are and others are not. But that's as far as we've been able to go with it.
Just following up on the deep sea fish, it would be brilliant to look at whether there's body form relationships to which types migrate. This extensive vertical migration. Some fishes do, deep sea fishes do it, and some don't.
Yeah, we do actually have a paper in review that's going in more detail in how this has evolved, and it's actually what the lead author of that study has found is that there's actually different periods of time that have led to the sort of invasion of the deep sea, and it's happened at two distinct periods either side of the KPG boundary, which is kind of interesting. But we haven't looked fully at migration. But good idea.
It looks like we have a question online. Ken, can you unmute and ask your question?
Yeah, sure. Yeah, thanks a lot for an inspiring talk. I was intrigued by your analysis of the streamlining. And I'm wondering if you also have measured the data on the tail alone, because there's very big differences in the swimming performance, whether the tail is tall and flat like on a tuna, or more like big and not very tall like on a cod.
Yeah, that is a really great question. So the closest we've got is we've got the caudal peduncle. So that's the part of the fish close to the tail that is holds all the muscles for thrust production. We don't have anything on fin shape. Obviously, fins would be really wonderful because, as you say, they're very strongly linked to locomotor performance. The problem is that they're also very sort of delicate and they get destroyed in collections a lot. And so we couldn't collect those traits from this kind of specimens, unfortunately.
Anybody else? Oh, okay.
So along those lines about, specimens getting destroyed, what's sort of the range of ages of the specimens that you were collecting the traits from, how long they'd been in the collections?
Yes, everything from a few years ago to 150 years ago. My students, when we flew out from California, they found ones that were like, wow, this is earlier than the state of California. So yeah, it's a whole range. And so some are in really good condition and others are not in such great condition.
Oh, very interesting talk. Can you hear me? Okay. Thank you for the talk. It's very interesting. Maybe I missed it, but I was wondering in what context did you use this kind of mean reverting kind of stochastic differerential equation? Was it like to look at the evolutionary process backward in time, or?
Yeah, so you're talking about the Ornstein-Uhlenbeck model. Yeah, so that's our model of trait evolution. So whenever we're looking at these traits over evolutionary time, we're using what's called phylogenetic comparative methods, which allows that model to model how the trait has changed along the phylogeny through time. So that's how we're using it.